a really rich, all right, we have a really rich collection of papers this week dealing broadly with the themes of biophilia and biophobia, delving into the realms of sourdough, microbial monsters, and also, you know, part of Rob's work is really about the microbial diversity of the human home and all of its complicated extreme permutations. Before the call, Salla and I were just talking briefly about, you know, how her foray into the art world has really generated some dialogue with broader theories of, you know, these kind of spaces where people make food in art spaces. So Claire Bishop's classic piece on antagonism and relational aesthetics talks about these classic ideas from Gramsci via Chantal Mouffe and Ernesto Laclau about antagonism being kind of a key part of a fully functioning democracy, where political frontiers are constantly being brought into debate where conflicts are sustained and not erased. And I was thinking, you know, as Salla is describing this figure of the microbiopolitical hacker, the hacker who's, you know, kind of reconfiguring her own microbial ecosystem, that we might think about relational antagonism amongst microbes as a way to think about, you know, this isn't just, you know, I think living in a pandemic where we're all afraid of these microbial monsters, the tendency is to treat all as enemy. And, you know, in some of the probiotic conversations, there's kind of the other extreme, like, let's welcome all the microbes into our gut and celebrate this happy existence. But maybe, like, you know, thinking with Claire Bishop and Laclau and Mouffe and Gramsci about antagonisms as being these productive parts of human social and political systems, maybe we could also import those kind of ideas into microbial ecologies to think with. So Salla, maybe I'll invite you to go first, and then Rob chime in. And Rob's also been doing some amazing new work that I hope to hear about relating to microbes in space. So yeah, there's a lot of things in the mix: monsters, microbes, space travel. But Salla, maybe you want to go first and say a little bit more about this paper and you know, how this relates to your broader work on probiotic communities.
Great, thanks, Eben. Thanks for the introduction, and I really think that the metaphor of the monster is quite an exciting one here. The paper that I've circulated initially came from my interest in taking part in fermentation collectives and fermentation workshops, but then sort of one part of my work is concerning sort of antimicrobial resistance and what it means as a social scientist to be working with environmental and clinical microbiologists to sort of think about the role of human practices in the tackle against the antimicrobial resistance. And some of us took part in a really cool fermentation workshop about a year ago now, actually, and I was invited to give a talk on fermentation and antimicrobial resistance, and I thought about that and I thought, there's no way I can put these two together. Like, they are just so far apart in terms of the way in which microbes are thought about in these fermentation communities, and then an antimicrobial resistance, which is often very clinically defined. It highlights the global health crises, how many people are dying. But the more I really thought about it, the more I thought that actually that antagonism is a really productive one, the different ways in which microbes are there defined, and thought about what they are seen to do, what kind of agency can they be seen to have, what they mean in a more politicized framing, was actually a really productive way of putting them together. And the more I then analyzed this fermentation workshop that I described with the sourdough bakers, actually they were also talking about microbes and how, antimicrobial resistance, and how the relationship with microbes was not just a matter of engaging with on some kind of biological terrain or a kind of earthly process, but actually it was a really much broader critique of serving as a kind of diffraction between lots of scales and bringing these scales together, of seeing the macrobial and the microbial, and sort of linking climate change and earthly destruction and the loss of biodiversity, political injustice, really just through that prism of living with microbes at the level of the sourdough. So it was a fantastic, that workshop in particular was a fantastic example of kind of making it, the fermentation workshop, a micro prism of these really broad political critiques that I really enjoyed thinking together. There's quite a lot there going on, and I will hope to continue the conversations, but I would like to hear Rob next.
Thanks, Salla. I was reminded how much I like to hear you think. So my work is in some ways super related and in some ways very, very distant. And so maybe I'll back out a little bit, that I do a lot of work at the interface of ecology and evolutionary biology in people's daily lives. And so recently, a lot of that's been about microbes in food and on bodies. And implicitly, part of the goal, and explicitly often, is to engage people in rethinking about their own lives, but also in thinking about science, thinking about their relationship to other species. And we've been doing this now in my group for for 15 years. And one more little bit of detail, which is that often the way we first engage people in these microbes is that we sequence them. So we identify who is present on this International Space Station or in your house or in a sourdough by decoding the genetic letters of the species. And so we can then generate a list of who's there. And about 5 years into doing this work, we had an encounter with the artist Reese, who does really amazing work at this interface of microbes and humans. And we were at the time doing work on belly button biodiversity, and so microbes and belly buttons, which was super fun. And she challenged us to think about, like, after we engaged people with their microbes and we gave them a list of their microbes and told them about them, did they then go home and have a better relationship with their microbes or a worse one? What did they do when they went home? And we realized we didn't know, and that nobody knew. And Joanna then did this intervention where she went out with some of the scientists in our group, and while having them swab their belly buttons, had them talk to their own microbes. And this was really radical. I mean, it was just wonderful. Well, first of all, people did it, which was amazing. But in doing it, they personified the microbes. They talked to them as fellow beings. And so something really interesting in that conversation happened that doesn't happen in our work. And so that's one little thing. And then the other thing is that we, like Salla, in a way, we both have some work that's about bad microbes and some work that's about good microbes. And we started to realize that when we were working on the bad microbes, like we work on microbes in shower heads, that the way people thought about them and talked about them when we didn't give them our language was very different from how they talked about the microbes in sourdough. If we gave them our language for describing the microbes, that overwhelmed everything, and that was the story, and we didn't hear very much. But if we listened, the bad microbes didn't have any embodiment. They were names to which these abilities have been ascribed, you know, and so non-tuberculous mycobacteria do these things, and were we to ask somebody to describe them, they would be round and bad, and that's it. But when we listen to people talking about their sourdough starters, if we didn't first describe the microbes and we let the people describe the starters, they described family members. And so the descriptions of the starters were really as these separate beings that they lived with, that were part of the family, that had their own histories, and that were described in kin network sorts of terms. And now I'm borrowing a work that Matthew Booker's done in collaboration with us. And so all of this has started to make me wonder, well, two things that I'd love to talk more about. One of which is, in what and how many ways have we failed by not doing a good enough job of listening to the ways that people are already describing the species around them? And so what ways do people have of talking about microbes due to their manifestations that I'm not doing a good job listening to because I'm throwing species names back at people? And so I've started to go through like some of the Scandinavian literature to figure out, when people were making sourdoughs, how did they talk about, you know, the yeast and things like this. I'm very interested in. And then the other is, would we be better able to deal with a pathogen if we attributed it to a physical presence like we used to, or like we sometimes attribute to monsters? If we thought of the coronavirus as looking like a monster, like that image that Evan shared, would we relate to it in a different way? And I'm really intrigued by these ideas. And I don't quite have the right framework for thinking about them, but I know that as scientists, we're failing in these conversations. We're failing to build on what's there in them. And then just briefly with where I'm going with some of this work, as Evan mentioned, we're characterizing some of the microbes on the space station and also some of the invisible animals that are for sure present. And so trying to think about how we talk about that living together that's happening, and of which the astronauts are not very aware, but they're aware of the aromas of these other beings. And how do you think about that? And so, for example, we just discovered a bunch of, how can I say this? I don't know. Imagine that there were tapeworms on the space station, and you might not be wrong. And so there's other stuff going on up there that's interesting. And the other thing is, I'm starting to work on how do we think about the fermentations that non-human animals do. And so wolverines take meat and store it. They store it in ice, they store it in water. And when they do so, they're actually manipulating the microorganisms there. How do we talk about that? How do we study it? Squirrels are doing the same thing when they bury nuts, and there's not a literature that describes this. And so it's brand new, and so we get to use the right way of thinking about it. And of course, I'm describing that from the vertebrate perspective, and we could also flip it around to reflect our multi-species ethnographies and think about the microbial perspective. And so I'm thinking about these sorts of things. And then also right now thinking a lot about our ancestors 2 million years ago and the extent to which maybe they were already engaging in fermentation. And what does that look like if early humans were not just bold hunters, but communal fermenters? And what is that story? And so, sorry, that's a little too much. I went too long, but those are things I'm thinking about.
Thanks, Rob. Super interesting stuff. For starters, there's a fun paper a friend of mine wrote about 20 years ago about fruit flies as fermenters that go around and see, you know, when the fruit fly is eating something, they're less eating the food, but they're eating the little bits of fungi that they seed on the food. But I've got a question about monsters. Going back to, you know, Mary Shelley's Frankenstein, one of the classic monster stories, you know, in the pop culture editions, like the movies and stuff, the scientific creation is the monster. But if you go back and read the story by Shelley and some other, like Mel Brooks' Young Frankenstein, kind of the lampoon Hollywood version of it, it's a story about the monstrosity of science and the monstrosity of society. And I wonder, you know, in the specificity of the figure of the antimicrobial resistant strains that Salla is talking about, or the monstrous coronavirus that's forcing us all to radically change our lives, I wonder what these invisible bugs tell us about the monstrosity of contemporary life.
Mm-hmm. Yeah, I think that's super interesting. I thought about with climate change too. If we personified climate change as a monster, implicit in that personification is the idea that behind that monster is the sociopolitics that enables the climate change. And so that to me seems even more like the resistance is like that too. Yeah, that's really interesting.
The antibiotic, when it was discovered almost 100 years ago, it was seen as a solution to what was at that point seen the monster, which was the microbe. So if Eben, you could share that image that that we had about the woman looking at her tea made from the Thames River water that was kind of teeming with bugs, teeming with microbes. It was sort of the advent of the discovery of the microbes, and it was the way that the public perceived microbes was sheer horror, that, oh my God, you've got all these things lurking in your tea. And it was just a metaphor for lurking absolutely everywhere around you. And the antibiotics then became the solution, and they've been praised as the miracle medicine that has made Western medicine what it is now. They are used by everybody all over the world. I mean, even in places with the most non-existent infrastructures, you will find antibiotic tablets on the market, on informal markets. And this is part of what the kind of research that I've been doing in very poor parts of West Africa. And so what is considered then the monstrosity in that story of antimicrobial resistance shifts and changes depending on what the available discourse, what the scientific discourse is, what the technologies are available, and so on. So the first in this photo, the bugs are the monster, and then the antibiotics and the public health intervention become the hero. But of course, fast forward 100 years, it becomes the antimicrobial resistance becomes the monstrosity, because that is indeed killing hundreds of thousands of people in Europe in and of itself every year. And so the public health perspective became a very dominant one regarding microbes when the microbes became pathogens that caused so many deaths and infections and human suffering. Were actually something that was supposed to be targeted, and then the definition of microbes underlying that public health approach was that they all must be bad. But and I would love to hear Rob talk more about this, but when you said, Rob, that you would now do the sequencing, now these more recent metagenomic methods that don't just rely on culturing and looking under petri dish, but actually can see much more in terms of the microbial diversity than what you can see with the culturing method. Actually, the scientists have realized that microbes have lots of different roles. There are much more of them than we even had any idea, and that they have very mutualistic, symbiotic, and important roles for human well-being. That's where all this new microbiome research comes in, and that our attempts to roll them out have created this new monstrosity for human and animal suffering, and indeed in ecologies more broadly. And so it's not a straightforward or unshifting narrative, but keeps changing, developing the kind of knowledge that exists.
Yeah, I mean, I think it's, yeah, that's a great insight. And you know, just this week or last week, Novozymes released a commercial cleaner for houses that they want to scale out massively. That it's microbes in a jar, right? And so this narrative is shifting all over the place.
Yeah.
And it's, yeah, the truth has always been a mix, but we're bad at things that are sometimes monsters and sometimes good, right? That's the extra hard part.
We don't always know
Yeah.
which one is it. Yes. So what do you make of these microbial cleaning products? There have been some around, perhaps used more in sort of marginal communities, not sort of, but because the mainstream has been these antibiotic sort of cleaners where you blow out all the antibacterial stuff, whereas some communities have been using these microbial sprays or cleaning detergents, not cleaning products rather, in order to create more resilient microbial climate or environments in houses that would then prevent the bad microbes to take over. I was wondering about that when reading your microbial divest in extreme habitats in human homes. Like, is there any point in adding more microbes to your houses? Is there an added value, or is that just 20 euros down the drain effectively?
I think it's potentially useful, but it's unregulated. And so there's not a good way to tell if someone's product, for the most, well, it's regulated very, there's a lot of topography to the regulation in different countries. And so it's very difficult to tell if anyone's shown that the product that you're using is beneficial. And I guess there are two parts there. Like, I would say vinegar, if you're using traditional vinegar to clean your kitchen, it still has live microbes in it. And so, that's actually a very similar approach and seems to work in some contexts. But I think that the secondary hope is you're actually getting microbes to colonize and grow and then do their job. And I think in a spray that then becomes more difficult. And so I think it depends on what role you imagine they have. Are you using their product? Are you using them in you're spraying them, you want them to do their thing for a very short period? Or do you imagine a carpet of new microbes that you've sprayed around your house and that cohabitates with you and keeps doing their service? And to me, the only place we've done that well is in the context of foods, that the sourdough, that's your food carpet of good life. But imagining that they're going to stay on a countertop or a cutting board, I'm initially skeptical, I think. But they'll become very popular very quickly. They will sell because it's very easy to market. That's my sense.
So thinking with Heather Paxson again, microbiopolitics, where every microbe is a hero or villain based on sort of situated effect and action and context. Scaling beyond food to think about the environments in our home, extreme or not, or the space station, which is an opportunity to sort of play with a petri dish of microbes and introduce good and selectively remove ones that are judged in a situated way to be bad. What kinds of maybe to pick a phrase that the three of us talked about in Sintra. So the three of us were at this workshop on fermentation, as they mentioned in the beginning a year ago. One of the phrases that emerged out of that is the phrase from ecology: intermediate disturbance. Is what you're looking for in a forest to create sort of a convivial biodiversity. So what kind of practices of intermediate disturbance in your household or on the space station, or what other kinds of tactics might we be thinking about beyond cooking? How do we sort of cultivate a convivial space that is good for human life, but also good for others in a more expansive sense? How do we think about the human as a holobiont, to bring in another keyword from biological theory?
So, Salla, do you have thoughts?
I have lots of thoughts on this, and I think much of the existing social science literature on sort of on these relationships seems to it suggests that there's been a trend in society more broadly, or in people's minds, and or sciences that has been abiotic or kind of antagonistic towards bacteria or microbes. And now that we are, with this new knowledge and this emphasis on these probiotic practices, we're undergoing something like of a probiotic turn. I'm not quite willing to make the picture quite as simplistic as that, in the sense that I think that there have always been lots of different ways in which microbes have been conceived, and that it's important to pay attention to which group of professionals is kind of lobbying a particular viewpoint. So depending on whether we are talking about infectious diseases doctors and their kudos and their development of professionalism historically, or nurses, or we talk about vets. And so I think a historian of science would see that the definitions, or the way in which different people or scientific professions are talking about microbes is very much connected to kind of their agenda of their power, what kinds of stakes are they trying to claim, what kind of knowledge production tools they have available for them. And so it's too simple to say that these dualisms can describe abiotic, antibiotic, or probiotic, and in fact there's lots more going on when you scratch the surface a little bit. And on my part, I like to think on more along the lines of practices, because if you then look at ethnographically, empirically, how people think about microbes, actually, like Rob, I think it's very productive not to say, well, tell me about what you think about microbes, but somehow approach the topic via something else. And so, hey, let's talk about fermentation, let's talk about thinking about sourdough, let's talk about your composting, and then the practices, focusing on the practices, actually unravels the story much more in often relational ways, but that the microbe there isn't it doesn't get singularized, but it allows a kind of multiplicity of different kinds of notions for microbes, depending on who's doing and what is doing and with what tools. So sourdough bakers talk about the flour, and they talk about the starter culture, and they maybe talk about their hands and the smells, and whereas bokashi fermenters also in a quite sensory way, but it smells like this, and then soil becomes like that, and this is how many critters I have in there. So in some ways, maintaining a multiplicity of notions of microbes for me is a very productive way to fall into these traps of dualism. And that also chimes with the ways in which microbes aren't just good or bad. They shift and change. E. coli can be completely mundane, and then it can be pathogenic depending on in whose body it is, or some microbes, like the coronavirus, seems to have been quite virulent and quite good at spreading, but bad for whom? So in some ways the multiplicity is also a condition of the microbes themselves. They can spread, they can give genes to one another, so the singularity on that level also doesn't really work by definition. So they exchange genes between species as well as within the same species. So I find that for a social scientific exploration of microbes, I find that it's worthwhile focusing on the practices, but then they're also maintaining the multiplicity notion about microbes, which all life scientists, this is completely self-evident, but it often, certainly in the popular view minds, the microbe can become something like it just means one thing, and I always am reminded that is obviously not the case, but it's for me productive to look at the practices, and which allows me to see the multiplicities of the definition that go from across all sorts of levels. That's a very long answer to your question, Eben, but I think this is the way I've thought about it. Rob, you're muted.
Thank you. If I can build on that a little bit too, then go back to the space station. So the work that has been done in the space station to date has shown that it has lots of extremophiles. So it has a lot of microbes from the astronauts sort of falling apart. So skin microbes, fecal microbes, oral microbes. And then it has microbes that live on the space station itself and degrade its parts. And so this was especially true on the Mir. The windows on the Mir actually clouded up with fungi. And so there was a time when the cosmonauts could not see out, which is a beautiful metaphor for some great work by somebody at some point there. And so you have these microbes there, despite everything being cleaned so much, despite there being so much focus on keeping these cosmonauts and astronauts healthy. And yet I still feel like there's part of me, and this goes to my own multiplicity, there's part of me that thinks, well, you're in an environment there where being sick is so costly that maybe it's still an environment where you try to control everything. And on the other hand, I know that the evidence is that that's not been possible. And I had this unique experience that I was giving a talk about this work in Mexico, and the guy stood up in the back and he said, I designed the filters on the International Space Station to control microbes. Are you telling me I shouldn't have? And I don't know. I don't quite know how to think about this. And so on the one hand, and so it goes back to Evan's question about the intermediate disturbance. Like, how much do you want to control these communities? How much do you want to lower their biomass? And so I think it's going to depend on the context. It's going to depend on whose perspective. And there are these cases like the International Space Station, like hospitals, and maybe like kitchens, where those multiple perspectives and their discordant conclusions are heightened. And I think those are really interesting places to think about these meanings and microbial consequences, because a lot is at stake and the environment is sort of circumscribed. And so what do you then do? What do you do in a surgical environment? And so I find that very often scientifically I'm without good answers in these situations. There's this whole ecosystem, and how do you best manage it when so much is at stake? You know, if you could design 10 different space stations, what would the experiment be that you would do, and how you would manage those microbes? And how would you want to think about it? I don't know. Go.
No, I think this is really important, because the staying with the multiplicity seems to be what is needed right now. So when we were alluding to the the dualisms with which microbes are discussed, it seems at the moment like there's an attempt to trying to get some kind of low-hanging fruit, easy answer of like what it is, that goes from someone asking, so are you saying that we shouldn't use soap because that causes depression? No, it's not so straightforward. Or like, should we be washing hands or not? Give me an answer. I think the fact that right now there's more uncertainty than certainty, and this is also very relevant during this COVID time, that actually what's kind of needed is to push back, trying to find some single or easy solutions about, or answers or definitions, but that kind of to accept that the knowledge is constantly evolving and changing, and that there's so much we don't know yet. These metagenomic methods have really shown that there's just so much uncertainty about it, and the way in which industrialization has changed so many ecologies that are erupting, so many other dynamics that involve microbes that we simply don't know. And I don't know, I would love to hear what people think about this, but somehow our human sense of ability to withstand complexity is what is at stake, because there's so much going on.
Yeah, and I think a remarkable feature of it is that it is unlikely to change in the next 1,000 years. Yeah. We are too big relative to them. They are too diverse relative to us. And so I think it will long be true that our ability to manage and understand is sufficiently clumsy that there's always going to be this uncertainty about that relationship. And how do you manage that well? And so the persistentness of it makes it, I think, all the more interesting to try to figure out how to do it.
Yeah, I mean, I know that Rob and I could go on doing this for a long time and then throw Eben into the conversation. We'll be here for the next 4 hours, I'm sure. So would anyone like to chip in?
Looks like there's a question that's just emerged from Natalie in the chat. Do you want to ask it out loud, Natalie?
Yes, I am really interested in that too, because I am working with cutaneous microbiota. That is really part of our identities. And yes, and about soap, and what is really great, I found extremely interesting, is the relation that we have, because it's in a strange way, our microbes communicate between them and with us. And that's why I would like to have your idea about that communication. Hello?
Yeah, no, that was my thinking face.
Excuse me.
About the communication.
Well, your example of the hands and the sourdough was such a great example of that connection.
Exactly.
Yeah, and what we've not done, and I actually think the sourdough, that it's very common that there's a way in which, so we happen to see it in the sourdough. It's not what we were studying, but we found that the bakers infuse the sourdough with their microbes, but also their hands change. And my guess is if we study people that embed their hands in things that are microbial every day, that if we looked at their bodies would change very often. That I think butchers are another good candidate. That my suspicion is that butchers have some of the common yeasts associated with curing meat.
How do their hands change?
Yes.
Yeah. Oh, sorry. Go ahead.
I want to know more about how their hands changed, or like if there are examples of how hands change and going from bakers to butchers.
Yeah. So what we found with the bakers is we brought a bunch of bakers together to make the same sourdough starter, same ingredients, and what varied, it was their air around them, and then their hands. And then we studied their hands, and then we studied what was in the sourdough that they made, and then we studied the bread. And what we found was, what we were trying to study was, do they convey microbes to the starter? And yes, they did. And that also influenced the flavor. But that was a modest effect. It was statistically detectable, but modest. But what we hadn't expected to find was that when we compared the baker's hands to hands of other people we had studied, that they were covered in 20, 30, up to 70% lactobacillus bacteria. And so the bacteria of the starters. And so it looks like what's happened is, from being in the bread every day, that they've actually have this alternate, we would call, ecologists speak, as alternate stable state, where their skin has then changed. And I mean, going back to Natalie's question, I mean, if we study, there's a group in Leipzig that studied aroma in chimpanzees and gorillas, and has found that chimps and gorillas both use each other's individual aromas in communicating. And so, you know, I know your smell, that smell is associated with this meaning. And most of that is produced by microbes on the body, fed by glands in the armpit and the perianal and vaginal regions. And so those aromas are markers of who those individuals are. And so in that way, going back to Natalie's question, when these bakers' bodies, when their microbes change from their daily actions of being in the bread, in some real way, they have also changed. They've become someone different, to the extent that a change in aroma marks who they are. And probably immunologically, they've also changed. And I mean, and I'll say from the bread perspective, we know this much about it, right? We have this little window, but that window is pretty fascinating. And so my sense is that if we were to look at other people who had as a part of their daily practice, immersing themselves in, farmers would be another group. If you're really putting your hands in the soil, the likelihood that that changes is pretty high. But it has to be, as Natalie suggests, a daily act, because just washing your hands doesn't change your microbes. So it really takes this. And so to me, the other sentiment that came home to me was, what do I want my body, my kids' bodies, to say about the lives that they've lived, about their communities of microbes, about how they've engaged nature and the world? I don't have a great answer to that question, but it was prompted by that thinking. Oh, and Sevgi has another. Oh, so does Kevin. Oh, hush.
Question of, I have a direct connection, but to the hands, which is in the paper. But Sevgi, do you want to address the immediate question?
Yeah, exactly. First? My question is, can you hear me?
Yeah, we can hear you.
Great. Directly about this, Rob, as far as I understand, in your study, you used the standardized protocol that you haven't, you did allow to use the sourdough mother, which is crucial and intergenerational, which we see in the Salla's paper. You haven't, you didn't allow them to use these sourdough mothers, their traditional cultures.
No, and it made them so mad.
Is it right? Yeah, I know. But it is my question. What is your insights if they would use these sourdough mothers? What do you think? How your results would? Probably it will be much messy, but I would like to hear what you think about that.
So, well, and I want Salla to jump in too, but before we forget with how this relates to her sentiment in her paper. But so one thing we do know is that some of the really old starters, so we've studied old starters through time, not through time. We've studied cases where an old starter was given to many people, and then we can compare those different descendants of the old starter, and we see that they're very much alike each other. And so right now it looks like those very old mothers may have a strong contribution from the original mother maker that then might be altering the people who work with it every day. And so there could be this familial back and forth between you and your, great grandpeople's mother that we didn't study, but is studyable. But also not allowing the bakers to use their mothers that they wanted to use. They were so mad, and which is also interesting. It was like something had been broken, that they couldn't connect with their way of making bread. And so they then snuck ingredients in with them. And so there was an amazing Turkish baker, and suddenly there was garlic and poppy seeds on his bread that was supposed to have the same ingredients as everybody else. And so, but there's something about this disruption that matter a lot to them. But Salla, you were going to say something, and then we have lots more here.
That was hilarious. Like, how did that change your protocol? Like, excluded sesame seeds? No, no. But the workshop that I wrote about, it had a huge significance for hands. The participants kind of self-organized themselves in this way. It wasn't necessarily something we did as a sort of evidence-based intervention. That people who were kneading the dough, they were mixing the starter culture and the flour and the batter. Some of them just did not want to wash their hands afterwards, and then they hang out for the rest of the workshop with like these crumbles on their hands. And that was, it was for them a way of sort of not just getting the microbes in through them with the bread, but actually like spreading the sourdough microbes into the room and touching things while they were then coming back to the barrel and kneading things. And what's so interesting that then in the conversation was elaborated that the sourdough was in some ways a vector, or the hands were the vector for connecting the microbes from the outside to the inside. And it was really important to them that the kinds of ingredients that they had in that overall process, as I write about it in the workshop, in the paper, is that they wanted to have organic flour and everything that was sort of according to their values of what was, I won't say use the loaded term pure food, because it wasn't pure in the sort of 18th century white flour polished out of not include sand and dirt, but rather with sort of moral values that Alexis Shotwell talks about purity in terms of the very, you know, wholemeal, organic, biodynamic, etc. These kinds of values. And it was that then the bypassing of the modernization of the industrial food production and an opportunity to strengthen the human environmental relationships through the selection of the kinds of flowers were used, that there was a whole line of kind of the macro-political to the micro-biopolitical, and that precisely worked through the hands, that they saw that the hands acted as the mediator between the kind of the politics and the microbes in the sourdough. And then when I read your paper, I was just like, they were onto something. They really were onto something, that the hands had played a role. It wasn't just a metaphor, but the hands really do have that connection of the microbes in the sourdough and what comes out of the environment around you in the kitchen or workshop.
Did Angelica, let's see,
Can I jump in with a question on this note? I'm curious, and I'm sorry if there's some background noise, but on this question of, as we're thinking about hands and microbiome and the microbia that live on our hands and how they shape us and sort of we shape the things that we make with these microbiomes, I've been thinking a lot in COVID times about like hand sanitizer and the role of sort of this, of like sanitization, and how like hand sanitizer and like this consistent sanitization and hand sanitizing might change those microbia and change that microbiome. Or I'm curious to hear like reflections on like what might the long-term effects of this kind of like sanitization intervention, are we risking something with the turn to like the constant use of hand sanitizer?
So I'll go back to Salla's multiplicities, which is that, so I just went to lunch with somebody who works on Helicobacter pylori, which is a stomach bacteria. And 1,000 years ago, most people would have had many strains of H. pylori. And it's thought that the way people get them, because they're not fecal oral, they don't live in your gut, they just live in your stomach, is mothers chewing food and giving it to their kids, or maybe communal food dishes that have some sort of saliva in them. And so H. pylori strains have actually been lost now in, say, in the US, for example, that a much smaller proportion of people have H. pylori. And some of the H. pylori is bad news, and some of the H. pylori is super good news. And so here's the multiplicity, or here's, and maybe I'm not using it right, Salla, but here is a complex component of this kind of loss that's associated with the changes in hygiene, where on the one hand you're losing some of these strains that benefit us, on the other hand you're losing strains that were bad news. And so how do you think about this? And I think the same thing happens with a lot of as we move toward like hand sanitizer sorts of things, where you have the potential that you're losing some of these species. On the other hand, you're balancing not getting some worse species. And so it's this complex thing that won't have a simple answer. But then the other piece that's interesting is there's the loss from an individual. There's like, if you lose, Angelica, one of your good microbes, versus there's our collective loss, that it just disappear entirely. And so in North America, there are a bunch of microbes that used to be common on skin and in guts that seem super rare now. And so they're like a collective loss, which seems like a different kind of thing from a personal loss. And how do you think about those?
So Julieta's just taken a phone call, it looks like, but she's got a super smart question in the chat about microbes as carriers of history outside of language, and inviting us to think about the gut microbiome, or the gut-brain axis, and how we might think about experiencing joy in these intergenerational collaborations as we cultivate these starter cultures. So Rob, do you know of anyone working on starter cultures and joy?
No, but that's a beautiful question.
And then situate that in a sort of historical knowledge or context, like in some kind of evolutionary context. Do we know anything about how those fibers would have been connected to serotonin back in the way in which those connections were produced in the human history? I don't know. I'll leave that to you.
I mean, so one thing we do know is Katie Amato, who is also in Sintra, has pointed me to a paper showing that there's a lactic acid specific receptor in human guts that underwent strong evolutionary change like 1 million years ago, potentially associated with eating fermented things. And as far as I understand it, we don't understand what that receptor then tells the brain, but it tells it something. And so that might be one place you would think about it. But it's also maybe that's too elaborate, because maybe in these communal foods, there's also something that's more like grooming, is when you're working physically with the food, is it more like chimpanzees grooming each other and that serotonin, because you're relating, if the sourdough is another human to you, if it's another being and you're grooming it, and maybe there's some physical response to that, just this act of engaging it. But it's a beautiful question. I'll be thinking about that for way longer than now.
And I think your answer points to the fact that we should resist sort of reducing the answer to any particular part, and not say, oh, it's not because of the biological connection, it's not because of the social connection. It could be all of that together in some ways, like all of the explanations of the grooming, the being that together, the way if it affects our genome or metabolism, whatever, they could all work together towards the answer, and not one or the other, or one of them over the other.
I think another layer of that would be aroma learning. So we now know that aromas don't have for the most part, like an inborn valence, like one aroma is not good versus another in humans, as far as we know so far, that we learn to like some and dislike others. But the ones we learn to like, we then do get a serotonin burst from those. And so you can imagine that as you learn to love the aroma of these foods, that is bodily, that there's something there too. Yeah, that's a good question.
I was just going to say, I really like that idea of grooming. And, we might think about grooming the sourdough culture, or grooming the home, or grooming the space station. And it reminds me of a sourdough artwork that my friend Jake Metcalf offered up to the Multi-Species Salon about a decade ago. It's called the sourdough culture that crossed the Oregon Trail and then started spreading itself on the internet. There was this guy in 1995 that put up a rudimentary web page, Carl, and he basically just made his family's sourdough culture available for anybody who wanted to send in a self-addressed stamped envelope. But Jake, in working with various sourdough cultures, found that some just went off. Some people who were celebrating like their local microbial terroir, just like letting the spore rain enter their culture, found it just didn't taste good. So, grooming the culture might mean just like categorically, and obviously people have been doing this for generations. Like, this one doesn't taste good anymore. This one smells bad. But there's still, like, hippies who are so connected. Like, I want my local sourdough culture, come what may. But I think that idea of grooming goes well with Paxson's microbiopolitics. Just thinking about the situated judgments we all are making. Whether it's like how many times we sanitize our hands in a day, or how much vacuuming we're going to be doing. It's all kind of a grooming process.
I think that's great. And I think like we had an ungroomed experience that I alluded to earlier, which is that we tried to do a big sourdough experiment in our lab, and it worked in the narrow domain of the actual experiment. But the normal yeast didn't colonize it. It was colonized by weird fungi. And so they still became starters, but they were terrible. And what we think happened is it's not a lived-in environment. And so it doesn't have the accumulated, even a small portfolio of accumulated yeast with which to work. And so you got these weird starters. And so the idea that people who make bread again and again are grooming themselves, grooming their family, grooming their homes, something that's not quite right, but something in there is very nice.
Yeah, it's a different, like, care is kind of the dominant feminist science and technology studies idiom right now. And I think adding, like, complicating care and thinking, like, there is, like, nitpicking is a particular kind of primate care that, we've inherited. And there's other forms of care, that we might talk about. There's a number of questions still in the chat, but one broad one that Salla and I were talking about a couple of months ago when we first had the idea for this event is, what do you make of all the prolific sourdough bakers in this time of the pandemic? I know that Salla has some of her thoughts, but this is being reported in The Guardian and other places. A lot of my friends are suddenly, investing a lot of time and energy in learning how to bake sourdough. Like, what does this mean?
I want to hear Salla think about it. I'd also like to hear Sevka think about this, because I think this relates to a lot of work that Sevka's been doing. Salla?
A very simple answer would be that it's almost it's not just about this Danish word hygge, where you're homely and comforty, but I mean it is that, but in a context of pandemic where a monstrous coronavirus is causing havoc, and then you want to maintain some kind of, safe connection with the microbes that and allows you to still feel like slightly, homely and hygge and self-sustainable, and not go out to buy things, but being self-reliant and strengthening kind of caring relationships at the face of a very pathogenic notion of bacteria. That is how it seems to be in, like, certain kind of biophilia at the kind of face of a more dangerous virus, while still not collapsing into a hopelessness completely and entirely. That allows you to feel a little bit safe and externalize the wonder of change as you can observe it and sense it in the sourdough when it blooms. The smell that Rob described a moment ago, the smell of freshly baked bread is just, you put that in your home, and even estate agents say that, brings bread smell, have bread smell in your house before people come and observe it. You're more likely to get it sold there and then, because it does something kind of profoundly, deeply comforting to people. And this might be all kitchen psychology, no pun intended, but this is how I've sort of very sort of psychological way of thinking about it. What does Rob think about that?
No, I like it. I also think that it's loneliness, right? That people want, that there's something, it's similar sentiment, but I also, one of the things we get a lot, because we engage a lot of people with their sourdoughs, is that people feel as though they've made a being, but they don't necessarily like that being. which is something interesting. Like I made a sourdough, and my sourdough is an asshole, and like it's needy, it smells terrible, and it doesn't make good bread. And so there's also that, right? The want to make something, the want to be, and then the feeling of like I made something not good.
A living Tamagotchi.
Yeah.
Yeah.
Let's see, does anyone want to have the last question or the last word?
Not really the last word, but I posed a question scrolling up in the question section. So we've heard about very benefiting relations to microbes, and I was wondering also, because Salla mentioned it in her paper, it was a play between vulnerability and nurturing or caring or grooming your microbes with these biohacking practices. And I was wondering, because it sounded very much like you needed to have the right equilibrium of microbes so that they would be really nice to you and, you know, you wouldn't get ill from them. And I was wondering whether you think that there might be the same notion of equilibrium and grooming and caring for your viruses, because usually we tend, in this multi-species context, we tend to exclude notions of vulnerability and notions of precarity as well of our own bodies, you know, with this being permeable to all kinds of microbes and viruses. So I was wondering whether this is a very specific thing for microbes, like microbacteria and yeast as well, you know, like fungi. So I was wondering whether what your thoughts were on whether you could actually transpose this to viruses as well.
So one quick thought is that the easy answer is in the context of bacteriophages, so viruses that attack bacteria. And so we know that like most species on your bacteria on your body have a specific virus that attacks them. And there are now lots of medical efforts to use those viruses to control those bacteria. And so it does extend to those viruses. But I take your question to also mean, how do we think about the viruses that are clearly bad to us in that equilibrium? And I have to think about that one some more, Kevin, but it's an interesting challenge. Like, how do you incorporate coronavirus into an equilibrium? I mean, is that a, yeah, I don't know. Salla, do you have thoughts?
I mean, there are, in addition to the phages, there are some viruses which are mutualistic or symbiotic that are necessary. It is generally viruses. I think they are thought of as non-living entities that then multiply and are pathogenic. But there are some symbiotic ones as well. Rob would probably know the literature better than I do. But I think the important question is like, if equilibrium refers to some kind of equal benefit, then I mean, I think it just really matters how do you think of the nature of the relationship. Like, is it in somebody's advantage, both parties benefiting mutually? Is it going one way, or is it the danger going one way and one way is net neutral? I think those in the literature on philosophy of science have been sharpened, but they're not. I think that is also the theory that has been questioned, since that it's not, those are kind of crystallized typologies, and often the reality between different species in those relationships are much more complex and complicated. And so the virus, perhaps from human perspective, in most cases of most vira, is perhaps a pathogenic one, but it like I said, those definitions are very they are very contextual and dependent on the nature of the relationship, whose perspective you're looking at, and so on.
May I just
go,
because
yeah.
no, just an observation. As soon as we talk about viruses, you're only talking about one species, whereas when you're talking about bacteria, you're talking about a whole environment of species. So I was actually referring to that kind of image, like you have various species of viruses as much as you have various species of bacteria, and those as well have some kind of relationship to one another, and we usually tend to exclude that. Like I was wondering whether you ever heard about anything, you know, like I know about influenza. There are all kinds of like supporting each other, but also some viruses exclude each other, and because obviously they if they populate the cell membrane, they might take up all the space, you know, for other viruses to jump in.
So yeah, I mean, that's a good question. I think part of it is methodological, like the way people have tended to study viruses has been individually. So you focus, you know, an individual researcher on the biology side would focus on some specific virus, and so you're the chicken influenza person, and so those interactions are like beyond your immediate purview. Vanessa Ezenwa has started to do research looking more at those interactions directly among viruses, EZENWA. But the other thought is that there's work on indirect effects, and there's more there. So a virus affects a bacteria species. If that bacteria species change in abundance affects another species, which then has a cascading effect. And so I think there's more if you include the viruses plus the bacteria plus the host. Maybe not always in a human sense, but there is more there. But the viruses are also hard. Like there's not yet been a single study of the viruses in homes in any sort of comprehensive way, not a single paper. And so we're just way earlier too. And so if you want to say, well, how many viruses are in your house? Good luck. There was a recent study trying to figure out which virus was causing colds in kids. And so they studied kids with colds and without colds. And the kids with colds had on average 5 species of viruses that could have been causing the cold. And so like it's just so early in some ways. Yeah.
Let's do that, Rob. In part, I love your kind of mass, crowd-sourced microbial ecology project. So I think it'd be fun to this group, we could launch our own sort of viral diversity project. So to be continued, but on a sort of philosophical and political in a political register, thinking about Kevin's question about viruses and equilibriums, there's Sophie Lewis's takedown of Donna Haraway's Joossens. So Donna is basically calling, I mean, there's a complicated thing going on in staying with the trouble of her latest book, where she's thinking about a moment after the present, a moment after this period that she calls the Great Dithering, where human population will be reduced to a much more friendly level for the biosphere. Some 2.5 million is 2.5 billion is a number she seizes on. But Sophie Lewis kind of tackles that question in terms of genocide. Like, if we're thinking about a dramatic reduction in human population, what does that look like? And there's been a lot of speculative fiction about this, where some well-meaning ecologically minded scientists in a lab designs either an experimental weapon in the realm of physics or a bioweapon, and achieves an equilibrium. And I think we also saw early in the pandemic where the Extinction Rebellion branding was hijacked by white supremacists saying, you know, this is the moment has arrived, you know, the reckoning, the moment of multi-species justice or interspecies justice. So I think we have to be careful in thinking about, yeah, the specificities of how these, like the ways that these are having disproportionate, this pandemic in particular is having disproportionate impact demographically, geographically. You know, as Tony Hatch talks about it, it's necropolitical warfare targeting kinds of people that are already experiencing dispossession and marginalization and inequality. So yeah, the question of equilibrium is interesting and is of critical importance. Like as we're living on this planet that not only is warming, but that is experiencing all kinds of other unraveling. But the ways that we imagine possible remedies and ecological imaginaries, I think is super important to kind of get right. But yeah, I don't want to kind of end on the note saying that Donna Haraway got it wrong, and maybe this is kind of a roundabout way back into monsters and a plug for my book, which has a final chapter where I'm sitting with Donna Haraway, reckoning with Sophie Lewis's criticism of her and imagining monstrous futures for humanity with CRISPR. But that was a plug for my book. But Salla and Rob, do you have any final words? We're officially over time.
I was thinking about the monstrosity when it's kind of preparing for this discussion, and it was so interesting when it occurred to me that what was the monstrosity kind of has shifted and changed over time. And I also came across a thought that chimed very much that had came from Margaret Sheldrake's work on the monster as something that provokes abject fear, and so it wasn't the monster in Haraway's thinking is very much the prompt to pay attention to materiality and earthly processes and so on, whereas the Sheldrake's definition seemed to still maintain on the notion of fear in a very human sense, rather than paying attention to sort of non-human or the multi-species, and yet it did something to me in terms of thinking about questioning dualisms, which to me in Western philosophy that and social theory that where our philosophical heritage is so imbued with dualisms of nature, culture, and society, the dead and the living, the man and the woman, the human and the animal, and I just like thinking with microbes, or just being with microbes or playing with microbes, where the emphasis is with microbes and not the human who is doing the being or thinking, to me it leaves me always completely changed. And I think it really questions the way in which social theory has been constructed through these dualisms, because like we talked about the multiplicities, we talked about how singularity is questioned because the microbes are in us, which are a part of us or outside of us, or with that, you know, are we the microbes or the microbes to us, and they can change genes with one another, so even on a cellular level they are not individuals, and we pass microbes to others through kissing, through touching, but also through the bread that we share and the food. And so our bodies are leaking in a way, in so many ways biologically. And so to me this is sort of the most interesting concept of thinking about all these multiplicities with microbes, precisely because they question so many of the ways in which classic social theory and Western philosophy has been built on these pure attempts to sort of distinguish the human and the different from the animal and the nature from the culture, and so thinking with Sheldrake's notion of the abject fear towards the thing that is being cut out in order to maintain something that is perhaps safe and contained, or the one versus all those that are other. I think the bringing the microbe back into the picture, not as the other, but like really as as a thing to think with, and giving it the agency that they have without thinking them through human concepts or having the human at the center point. To me, this is a revolutionary process for social sciences.
Final thoughts, Rob?
Yeah, I'm stewing on Salla's final thoughts. I think one of the things that's going to be rich for me to think about for the next years is how have people already come to terms with thinking about microbes that we've not reckoned with? And Sevga's work is really beautiful in this regard of thinking about traditional fermenters and how do they talk about microbes? How do they think about them? How do they curate them and groom them? And so I think that's a place where we can learn a lot, and we've mostly just failed to listen. It's right there. It's not far away. And I feel, and this is what I was trying to process with Salla's closing thoughts, that as scientists, we lack the right language right now for dealing with these multiplicities. And we're very bound by our fields. And so one group studies one virus, another group studies another virus until the tool changes. And Sevga shared a paper that's a virome paper, which is a reflection of the tool changing. So we study it in a new way. And then the other thing that I'm still stewing on is, are there still useful aspects of monsters and gods, let's say? And so is there a socio-ecological function to turning some of these things that are hard to think about into monsters or gods? And here I think back to sacred forests, that various places around the world, both sacred forests that are filled with traditional gods and dangerous forests filled with monsters have both been more likely to be conserved. And so here was this socio-ecological function of the monster that was put in the place that was not yet understood well enough. And so I'm interested in that idea, and so is there a monster or a god that we could position over the microbes as we come to understand them better? And Salla's thoughts have made me think that maybe that's too simple a way of thinking about it, but that's where I am, maybe pondering rather than concluding.
I think that's a great way to conclude. I love these monsters and gods as productive figures to carry forward. We've got a number of interesting other speakers coming up. Next week, Alondra Nelson talking about Black lives and afterlives. Lauren Carruth, who was just canoeing with yesterday, who's got some really interesting stuff on camels and MERS. I went canoeing with her yesterday, and she's also been working on camel milk and fermentation. So things that we were just talking about today, I think we'll carry forward towards that on October 5th. But yeah, thanks to both Rob and Salla. It's been a lovely opportunity to finally have this conversation. And yeah, let's sequence the virome of our bodies and homes. I think that would be, let's get some money and do it.
Fun,
Sounds
but harder
fun.
than it sounds. It's great to meet everybody. Thank you, Evan.
See y'all.
Thanks for the wonderful
Bye.
questions, everybody.
Thanks. Evan, Rachel, would you be able to keep a hold of the chat as well?
Sure. Yeah. We'll copy that and send it to you in an email.
Thanks.
All right.
Lots of really nice thoughts there.
Yeah.
Thank you for organizing.
Bud.
This was really, really great.
Thanks, folks.
Bye, everybody.
Ciao.
Bye.